It is my pleasure to introduce our next speaker, Dr. Annie Tao, who is professor of biochemistry and chemistry at Purdue University and is one of the two awardees of the 2024 Catherine Peachy Breast Cancer Research Grants. And we are so pleased also that our generous funding partners, Stephanie Lesher, I've seen Heather Pirovsky, I don't know, is Carrie Kanya Sola also here? From the Peachy Foundation are actually here with us today, so this is exciting. Dr. Tao will talk about profiling molecular fingerprints of breast cancer subtypes through plasma-derived extracellular vesicles.
So when I was preparing the talk over the weekend, so I tried to keep up with today's fashion. So what is that? Is I ask AI. So, so what does it look like for breast cancer diagnosis in the future? So here we go. So that's is the diagram, is the figure we eventually get. And so you can see from the figure, so this is generated by Google Labs. So you kind of do some measurement at home, and you get the parameters, and then apparently through cloud or something like that, you send it through remotely. And in the end, so either AI doctor or real doctor will be carry out the diagnosis remotely, and for you, so you don't need to leave. So I think depending on your angle, you think maybe how far away from this. I first feel this is seems like an ideal case. So AI more or less did a reasonably good job. And the second one is depending on angle, someone think that's still far away, and someone think that's pretty close. So, but at any rate, so I feel my lab today I'm going to talk about two quick projects actually toward this goal. And so both of these goals are based on this particular particle called EVs. So it's not an vesicle, it's actually called extracellular vesicles. So these extracellular vesicles has been discovered actually you can in all kind of biofluids such as blood, urine, CSF, or saliva. So these are the nanoparticle range from nanoparticle to these micrometer particles, and so it's essentially double-layer membranes. So it's more like small cells, except much smaller. And so you can find it in our biofluids, and then they are secreted by all the organs and every type of cells. So there are a lot of them in it. And in these extra vesicles, so there are nucleic acids and proteins and metabolites. And so these days, actually, a lot of the biology turn out to every biology you can think of, EV actually can do. So it's kind of something amazing, something also kind of confusing, right? So, and related to my lab's research, the first we found that take advantage one is that a lot of these proteins are functional proteins, they are post-translation modified. So these are not easily to find in the blood because they are not stable. Because they are being encapsulated in these extra vesicles, they can be quite stable. And so my lab has been working on these modified proteins, so we really take advantage of this feature. Second feature is the reason today's these blood-based and also urine-based tests still limited because it was very complicated. And then by bypassing all these complications, focusing on these extra vesicles, actually we can really bypass these challenges. So my lab is really taking advantage of these two major features. And so we have been working on these extra vesicles for about 10 years now. And so we use these EV sources from cell culture, from tissues, from blood, from urine, from saliva. And so all can see that. And then one major part is isolation with the EVs. So my lab really put a lot of effort into it. The current more standard is ultracentrifugation. But this method certainly is not very practical for some of these future usages. So my lab developed some of the chemical affinity-based method, including we come up with a magnetic beads called EV trap, which is commercialized about a couple of years ago. And then the third one is considered my lab's specialty, that's proteomics. So we carry out a lot of proteomics on these EVs, and in particular with modified proteins in these extracellular vesicles. And then in terms of disease type, we focus quite a bit on cancer, but for past several years, actually, we also work on a good efforts on the neurodegeneration disease, such as Parkinson's disease and Alzheimer's disease. So today I'm going to first highlight a couple of early studies in my group related to breast cancer research. So actually this was the first study come out of my lab on extravesicles. So in 2027, we reported actually really identify actually many more than I originally anticipated. Before we published this paper, from a blood sample, we typically don't see these phosphorate proteins. The phosphorate proteins certainly are considered very important signaling molecule in cancer, and usually they couldn't see in the blood, but by focusing on these extravesicles, we observe a lot of them. So this is the 2017 work, and then so move on to 2020, and so we have another study, but it's essentially a workflow, saying that especially we work with clinical samples, we demonstrate that by using same starting material, you can look at different modifications sequentially, one by one, look at, for example, glycosylation, phosphorylation, acetylation, all these studies. And so by highlighting these two studies, and so we at least in my understanding, I gave two pieces of information. Number one is that I always tell my students that my lab is not a biology lab, so we don't need to invent a new biology. What we want to prove is that some of these important markers, they have already been proven in the cellular level, in the culture cell, in the animal model, in tissue samples. They're already well studied. We only need to prove them in the biofluid so we can see them. That's exactly what we see. For example, here's the data. We see some of these oncogene as being actually really discovered in our samples, in extracellular vesicle samples. Second one is how we really couple these kind of knowledge with clinical samples and make it easy and potential translational in the future. So today I'm going to highlight a bit of study with my student. I would like to highlight that actually related to today's talk, that first study and last study is that Hilary, my former student who joined my lab, actually she came to me, she actually came from a family with a history of breast cancers, that including her grandma and also aunts. And so she was motivated to that, so that I immediately put her into this project. So she was in the middle of the co-authors, and then by the time she graduated from my lab in 2020, so she was first author for this Nature protocol paper. And with her drive and also motivation, so that my lab continue working in the breast cancer field. So one study I want to highlight today is that's about to look at the subtypes of breast cancer. So we want to demonstrate that not only be able to differentiate cancer versus non-cancer, but for subtypes. Because for this conference, I don't think I need to go into give a lot about subtypes. What exactly I'm showing that for today's, I think the breast cancer probably most studied cancer types, and so subtypes, they have different prognosis and diagnosis, and also the reason that depend on different subtypes, so the cancer treatment would be different, because some can be effective, some will be less effective for the different types. So we want to see that we'll be able to use the extravesicles to differentiate different subtypes. So we got a sample actually from University of Iowa because I gave a talk over there, and so unfortunately Purdue didn't have the medical school. I gave a talk in University of Iowa's medical school. Said they have pretty good biobank. So we get the patient sample from them, and then we from the Susan Cohen's group in Indianapolis get the control samples. And so total we get about over 100 samples. So we carry out these samples essentially on the 3 stages. One is essentially do the screening, and the second one is validation. Third one we do a bit more cross-validation with other studies. So the protocol we use is based on nature protocol, as I mentioned. You can use the same clinical sample. You can extract different modified proteins. We start with tutor proteins, and we can get phosphorylated protein, and we get acylated protein. We can get glycosylated proteins, so that we can really get a multiple layer of information. And we hope these multiple layer of information give us a more accurate fingerprint of the subtypes. So that's the concept. But I won't go into details. So in terms of data, so we get one of the most comprehensive profiles for sure in the EVs, particular for the plasma EV samples. And so we identified thousands of peptide phosphoproteins and glycosylations. So and here we can make the comparison across different groups. And so we do this called heat maps. As I mentioned, I want to get into details. What exact information get? I already mentioned it. I didn't try to identify new information. What I prove here, exactly information being reported by others. For example, we look at the acylation, we see the TCA cycles being upregulated in the LUMA A and B. And then for the glycosylation, we see the insulin-like growth factor, is a receptor, and also ECM, this glycosylation also being upregulated in triple-negative breast cancers. That's also being well reported, but we being reported in the extravesicles. And then the most information we extract from it is the phosphorylation. So we do see that phosphorylation is a lot of these signaling is really related to cancer has been essentially dysregulated, and including EGFR, including others. So these are certainly, we feel that a lot of information can extract from. For example, I take 2 examples with there's 2 proteins. I don't get into details. These 2 proteins being reported by multiple groups. They are upregulated, would affect the survival rate and all these. So we did see them and can differentiate them from the LUMA AB. So they are more upregulated in the triple-negative subtypes with that. So with this kind of information, we went back to do some validation, because these are we isolated from the patient samples. As I mentioned, every organs can release the extravesicles. How do you know these are from your breast tissues or these? So we really compare our data against these cell cultures cells using breast cancer cell lines. So as I mentioned, the breast cancer potentially most well-studied cancer, so that there are different cell lines for the LUMA AB and for the triple negative. So we compare against that. So we validate some of these, for example, upregulated phosphorylations. Indeed, we see also in the cell culture models. So in that case, these EVs are considered more pure because they only come from the cell types. So we sort of validate it. So after that, so we really want questions. We see the crosstalk between these, why we do all these multi-layers of modifications. And the major reason is that we see the crosstalk between these modifications. So I give one example of PD-L1. I'm sure a lot of you guys know PD-L1 being the one of the major molecules being today developed these antibodies against for immune oncotherapy, and most of the antibodies against PD-L1. And so it's because PD-L1 is compromised the immune basically response, and they usually bonding onto the PD in our T cells, and then T cell will not be effective. So in the PD-L1, in the extracellular domain, is glycosylated because it's a cell membrane protein, and the inside is phosphorated. And we observe the glycosylated indeed in the triple negative is being upregulated, increased, and then in the phosphorylation is downregulated, is precisely being reported and on these. So both of them actually will suppress the immune system. For example, phosphorylation, the phosphorylation, increased phosphorylation usually will degrade this protein, but downregulate this one, which means will increase the expression of PD-L, which will further suppress the immune response. So that's precise being seen and reported by multiple groups. We see them like that. So that's would be considered cross-talk, and we went ahead that using this approach and look at the target approach, and that target approach actually we see them actually really consider try to validate of them, and with that, so we carry out today's sort of majority work we start getting into it is the machine learning and the AI studies. Eventually we come up with panel, and that's including, for example, this glycosylation and phosphorylation would distinguish the LUMO AB from triple negative, and then phosphorylation and also the glycosylation distinguish triple negative from the LUMO AB, so which is machine learning with that, with this. So these are the work. So we basically look at subtype with that. So with show me only less than 2 minutes, I very quick give you another project. That's we come at how you can eventually do at home. So we come up with this dry blood called EV collection card. So you can get it from like in Walgreens or CVS, but except that is not suitable for all studies. So we made these prototypes. So here's showing already some of the figures. So essentially, so here's the diagram. So you do fingerprint. It's about 100 microliter of blood. In the end, using these membrane base, we have a multiple layer of membrane. In the end, you get the plasma flows through this membrane. And the last membrane we coated with EV capture this reagent. So that reagent would capture the EVs and also preserve the EVs. The idea is that the EVs is nanoparticle, is actually really relatively stable. So I show you the data later on. So after that, so you can put this card into the zipper bag and then you ship it back to the lab, and then the lab will extract the extra vesicles and carry any analysis you would like. And then from then you can get a nucleic acid, you can get a protein, and if you want, you can get the metabolites. So here's showing some example. So we give very much characterization of the EVs and showing that almost the same as we get from vein blood. It's very similar data. And in terms of get these proteins, we found also contaminant is quite low compared to that with that. So then the third one is stability. So we put it into dry up to 30 days. And the EV is stable, so indicating that you really can ship out and store for quite a while and without effect on that. So finally, we have the data. Actually, we have several individuals. Actually, we get with the IU Medical School on these individuals, and we carry out analysis on these individual samples. On the multiple analysis, you can clearly actually differentiate them, indicating these EVs preserve the original each individual's signatures. You can differentiate them, including someone with COVID-19 actually to stand out with that. So I stop here. I don't have the device with the breast cancer data yet because we don't have the IRB, so we need to get approved. Once we have that, we feel this would be most suitable for breast cancer patients, including, for example, my former student who come from the high-risk population of breast cancer. So with this kind of device, I hope that you can do every week or even more frequent for monitoring that. Based on last year, I attended the women's the symposium that we have a technology, just we don't measure them more frequently to catch that precise window. I hope this device would be able to at least address some of these issues. All right, so I finish here, and thank especially WGHI and the Peach Foundation for the support. And so here's my group. The reason I chose this was when my student, we attended a meeting in Houston and get this pink cowboy hat. I feel this appropriate for the meeting. Thank you very much.